I have already done this is fourth lecture on the protein synthesis, and where we are going to talk about the eukaryotic translation. So, similar to prokaryotic translation, prokaryotic ribosomal component ribosomes. In eukaryotes also we are having ribosomes that is composed of two different components. So eukaryotic ribosomes is composed of ATS, which we are going to see here. This ATS ribosomes is composed of two different components. One is 60S, other one is 40S. This ribosomes, as we are knowing that ribosome is composed of ribosomal RNA plus different proteins. So all together, these two combine together and give us ribonucleoproteins, which forms ribosomal component, and ultimately ribosomal components forms ribosome. So large subunit of ribosomes, that is 60S, composed of, it contains three different ribosomal RNA, that is 21,8, 28S, 5.8S, and 5S, and it also contains 49 different proteins. Similarly, 40S ribosomes of eukaryotic cells contains only one ribosomal component, that is 18S, and it contains 33 proteins. So this is the total means the components of the ribosomes of eukaryotic cells. It contains four different ribosomal RNA, that is 28S, 5.8S, 5S in the large subunit, and the 18S in the small subunit, and then it contains some of the proteins. This ribosomes, already we are knowing that there are three different specific sites. It is having, that is called as E site, P site, and A site. So this there are three. Means these site is having some specific specification, that some significance. This E site means it is an exit site. This P site means peptidyl sites, yeah, peptide binding sites, and this A site is amino acyl site, means the except the first amino acid, almost all the amino acid except first one comes first time to this A site. So that's why it is called as amino acyl site. So I mean tRNA along with the messenger means along with the amino acid comes to this A site the first time. This P site is the place where peptide bonding occurs. So peptide formation occurs at this P site. So that's why it is peptidyl sites. And the E site is the exit site from where tRNA, uncharged tRNA, means after the peptide bond formation, the free tRNA, which does not have any kind of amino acid, will exit from this E site. Other important thing in this part is this E site is only present on the large subunit. Remaining other two sites like P and A site contributed by both large and small subunits. So there some portion will be on the large subunit, some portion will be on the small subunit. Whereas in case of E site, it is exclusively contains 100% in the large ribosomal component itself. And these sites is having some specifications, some significance that we are going to see in further lectures. Already we are knowing that this protein synthesis occurs in the amino terminal to the carboxy terminal, means if this is the peptide which we are going to synthesize, so this will be amino terminus, this will be carboxy terminal, and protein chain is going in this direction. So the direction, like in case of DNA RNA, the direction of growth is always 5 prime to 3 prime. So chain is growing towards 3 prime direction. Similarly, peptide chain grow from the amino to the carboxy terminal. And this ribosomes reads the messenger RNA in 5'3" direction. That already I have told you. Here RNA will grow, ribosomes will move in this direction towards 3 prime ends. So chain of peptide synthesis growing on messenger RNA is 5'3" direction only. And there is a polyribosomes. It forms polyribosomes. Polyribosomes means multiple ribosomes on the same messenger RNA, which gives us multiple protein at a time. Total protein synthesis we are going to divide similar to the prokaryotic protein synthesis into 5 different steps. Those steps are activation, activation of amino acid, you can call it charging of amino acid, charging of tRNA, you can all call it. Then initiation, elongation, termination, and the post-translational processing. This is a modification of the proteins. So first we are going to see activation of amino acid. In activation of amino acid, the reaction occurs in two different steps. In the first step, the amino acid whatever amino acid we are having, it is going to react with ATP. So it will use AMP from this ATP, and AMP will combine with this amino acid and ultimately forms amino acyl AMP, and pyrophosphate will come out. So this pyrophosphate is coming from this ATP. AMP will be used along with amino acid. This reaction occurs in the presence of enzyme that is called as amino acyl tRNA synthetase. So for every amino acid, we are having a specific enzymes. So these enzymes helps in the loading of amino acid on the tRNA. Second reaction is this amino acyl AMP, which we have formed in the first reaction, is going to combine with the react with the tRNA. As we are already knowing that at the tRNA 3' end we are having CCA. This A is 3' end, so it is having hydroxyl group. So this hydroxyl group helps in the reacts with the this lone pair of electron of oxygen reacts with the AMP, and AMP will be dissociated and ultimately tRNA associated with the amino acid. So we are having this tRNA, here we are having amino acid. So ultimate reaction will be amino acid reacts with tRNA in the presence of ATP. ATP will be mediated or energy source, you can call it. Enzyme will be amino acyl tRNA synthetase, and this amino acid will be loaded on tRNA. Ultimately, we have got amino acyl tRNA plus ATP will be dissociated into AMP and pyrophosphate. This is the overall reaction. This is the first reaction where amino acid reacts with ATP. ATP means AMP plus pyrophosphate. So pyrophosphate exit in the form of two phosphate, and AMP will combine with ATP, and ultimately adenylated amino acid or acyl amino acid. We have got it. So this is AMP portion, and this is amino acid portion. In the second step, this amino acyl AMP reacts with this tRNA. This is CCA 3' end of tRNA. So this reacts together, AMP will be dissociated and amino acid will be loaded on the 3' end of tRNA. So ultimately, this is the activated tRNA, we have got it. So for every amino acid, we are having activated tRNA, means amino acid will be loaded on the tRNA. Now, there are two things we need to remember it. In the eukaryotic cells, the initiating amino acid is methionine, as it is also in the prokaryotic cells. But the difference is, in case of prokaryotic cells, we had formylation. But in case of eukaryotic cells, we don't have formylation. So no informal methionine is the first amino acid of eukaryotes. Rather than it is simple methionine is the first amino acid. But the two tRNA for first methionine and the other methionines, there are different tRNA. So the tRNA which is going to carry methionine for the first place, that we are going to call tRNA methionine I. I means initial, and remaining we don't have I subscript or superscript. So it will be for other things. So the first thing we need to remember is no formylation occurs in case of eukaryotic cells. Second is we are using normal methionine, but for both methionine we are having two different types of tRNA, and the loading on the both the tRNA, either it will be tRNA methionine I or tRNA methionine, loading will be done by same amino acid tRNA synthetase. Sorry, it will be synthetase. So the same enzyme going to catalyze the activation of tRNA in both the cases. That is the two different reaction, two differences in case of eukaryotes and prokaryotes here. Then we are going to see the initiation reactions. Initiation, in case of initiation, we have already seen that this is not formylation, this is AUG. In case of prokaryotic cells, we have roughly 8 to 13 base pair apart. We had a Sine-Delgado sequence. In case of eukaryotic, it is not Sine-Delgado, it is Kojak sequence. So this Kojak sequence is complementary with the not 16s, 18s, because this picture is on from the E. coli, so that's why it is written prokaryotic components like Sine-Delgado sequences, but we are having Kojak sequences. This binds with not 16s, this binds with 18s ribosomal RNA of 18S ribosomal RNA of 40S RNA ribosome, 40S ribosome. But the difference is, in case of prokaryotic cells, this binding helps in the AUG to fit at P site. But in case of eukaryotic cells, this is not the case. In eukaryotic cells, RNA binds, RNA, ribosomes bind at the first, uh, means the initial means first nucleotides of the messenger RNA, and then scans wherever they will get, they will encounter the first AUG, they will stop. That first encountering AUG will be the initial codon, yeah, initiating codon. So in case of eukaryotic cells, it is not done by this combination, this association. The first amino acid finding will be done by the scanning of ribosome. These are the different factors. These factors we will understand in the further slides, how where we are going to use these factors. So one of the factor which is ELF4 4A, that is having RNA helicase activity. The purpose of this RNA helicase is any kind of secondary structure, any kind of association between the self-complementary nature of RNA. So all those things will be digested by this 4A enzyme, that is RNA helicase activity it is having. So now we are not going to see all these things. We will see in further slides all these components, and in the last we will talk about all the components. Another component we have, the important component is ELF, initiation factor 3. E means eukaryotic, I means initiation, and F means factor. This is IEF3. This IEF is also having a significance. This IEF combines the 5' end and 3' end of the messenger RNA. So 5' and 3' associated together with some kind of proteins. One of the protein is IEF, means EIF, in eukaryotic initiation factor 3. This IEF has some other significance also. This helps in the assembly of ternary complex. We need to remember it. We will see what is ternary complex. Then the binding of ternary complex with other component, that is 43S pre-initiation complex. It will help. Then it will also find help in the finding of messenger RNA to this 43 pre-initiation complex, and scanning of messenger RNA for the AUG codon. So all these things will be assisted by this initiation factor 3. We will see what are those steps. So translation initiation in case of eukaryotic cells. These are the 4 different steps, 4 major steps of the initiation of the translation in the eukaryotic cells. The first is ribosomal dissociation, where 80S ribosome will be dissociated into 60 and 40S. That is, then 143S pre-initiation complex formation will be there, and then 48S, and lastly 80S. So this is the final 80S, means 60 and 40 will be combined together. This, there are 2 factors which we are which helps in the dissociation of 60 and 40S, which does not allow this 60 and 40S to come together, which dissociates them. They are called as anti-association factor or dissociating factor. These are initiating factor 3, just now we have seen, and factor 1A. So these 2 are called as anti-association factor. These 2 also stabilizes these pre-initiation complex, means give the stability to the pre-initiation complex. So now we are going to see different steps of the initiation reaction. So we are going to see first ribosomal dissociation. This is a ribosome which is having a 60S, which is having, second one is 40S. This ribosomes will be dissociated into 2 different components with the help of 2 factors. These factors are initiation factor 3 and IF1A. These 2 are called as anti-association factor. These 2 factors dissociates 60S and 40S. 60S is going to combine with initiation factor IF6, whereas these 2 factors, which is anti-association factor, associated with 40S ribosomes. This Then we are second part is we are going to form a ternary complex. This ternary complex requires some factors. Factors are initiation factor 2. It requires GTP. So these two combines together and gives us if effective a functional initiating factor. IF2 will be effective, will be functional only when it will have GTP. If it does not have GTP, it is not effective. It is not functional. So this two combines with the loaded tRNA. This is the loaded tRNA, activated tRNA. From the first step we are getting. In our case, it is methionine. So this is loaded tRNA with the methionine. These two combines together, this factor and these two combine together and gives us a complex where tRNA loaded with methionine plus this initiating factor. This is called as ternary complex because three components are there. One is this one, second one is this one, and third one is this one. This is called as ternary complex, and this is called as binary complex. So binary complex combines with tRNA loaded with methionine, and it is called as ternary complex. In the second reactions, next reaction we are going to form 43S. So we have seen four reactions. Four reaction means first one was ribosome dissociation, second one was 43S pre-initiation complex formation. So now we are going to see 43S pre-initiation complex. So we have got 40S. This is our 40S, which is having two factors, 1A and initiation factor 3. And then we are having this ternary complex. These two combine together and forms a complex. This complex is having 40S associated with EIF, initiating factor 1A, initiating factor 3, and factor 2 with GTP. This is called as 43S pre-initiation complex. In this pre-initiation complex, we are having factors, components are, this is 1 40S, this is anti-association factor, this is tRNA loaded with methionine, and this is binary complex. So this is called as pre-initiation complex. So in the further reaction, we are going to use this pre-initiation complex. So the one of the function of this one was factor I3 was it these two give the stability to this pre-initiation complex. So this complex stability will be given by this factor and this factor. So these two stabilizes them. So 43S equal to 40S plus IF3, IF1A, IF2, GTP, and methionine tRNA. So this is our 43S. Now we are going to see the messenger RNA. Our messenger RNA is having some specification. Those specifications are normally this is our messenger RNA, which is having poly A tail at the 3' ends. In our case, this poly A tail is covered with a poly A tail binding protein that is called as PAB, poly A tail binding protein. At the 5' end, it is having different components, different factors. These factors are 4A, 4G, 4E. All together, we are calling this factor as 4F. So 4F equal to 4G, 4E, and 4A. And this is called as cap binding protein. So at 3' end, we are having poly A tail binding protein. At 5' end, we are having cap binding protein. This normally by when we are going to see this messenger RNA looks like a straight, but it's not exactly straight. It will be this poly A tail binding protein and this cap binding protein will form a loop. This is the things. This is the loop. How it will be there. So exactly cap E, this for 4E associated with the cap. So that is cap binding actual proteins. This cap binding protein and poly binding protein will be held together by 4G. So this will be held together with this one. So E and PAB will be held together with the help of 4G. And another protein which seals them, that is EIF3, which already we have seen in that was part of our 40S ribosomal subunit. So our messenger RNA is normally in this structure. The next step of initiation is 40S 48s initiation complex formation. So this is our 43s pre-initiation complex, which is having 40s EIA1, 3, 2, GTP and tRNA. This combines with this messenger RNA and in presence of another factor, that is 4B. So 4B, and ultimately we are going to get a component that is called as 48S initiation complex. So the components with 48 8S is, that is 43S messenger RNA, along with in the messenger RNA we are having 4B, 4F, 4F means A, E and G. This 43S equal to 40S factor 3, factor 1A, factor 2 with GTP and tRNA. So 48S equal to, if we will replace this 43S here with this components, so this is 43S plus messenger RNA plus 4B, 4F and 4FA, all those things. So this is our 48S, which is having a number of components. So this is almost the final thing, except it is does not have a large ribosomal subunits. So now next step will be ATS initiation complex formation. In this ATS initiation complex, we are having 48S from the previous steps. This combines with this 60S ribosomal subunit from the first when ribosome was dissociated. So this ribosome was dissociated, and 60S will combine with IF6. These two combination needs another factor that is called as initiation factor 5. So what are the things we have used? We have used 1A, 2, 3, 4, all the four complex, 5 and 6 is here. So we have used all the factor 1A, 2, 3, 4B, 4A, 4E, 4G and 5 and 6. Once this once this 48 is combined with the 60S in the presence of EIF5, it forms a complex that is called as ATS complex, and all factors has been removed, means all the factor will be dissociated from there. So 1, 2, 3, 4, 5, 6, everything has gone. So ultimately we have got this factor, this is called as ATS complex. From this ATS, what are the things exit? That is 1A, factor 2, factor 3, factor 4, factor 5, factor 6. So everything gone. Now this is ATS initiation complex. In this ATS initiation complex, what are the things we are having? We are having 40S, 60S, messenger RNA, PAB, tRNA loaded with amino acid. This is our ATS initiation complex, which is going to initiate our protein synthesis. Now, this protein synthesis scans for the 48S scans. Just a second. This 48S, in this case, this scans wherever they will find AUG, they will bind. So that will be the first amino acid, first protein synthesis process start from there. So that is our first amino acid. Now, sorry, this 48S initiation complex bind with 60S. We have already seen, and form 80S energy is required for this, which is coming from the GTP, which was associated with the binary complex EIF2. This step helps by EIF5. Already we have seen, in as ATS forms, all the factors will be released. So in the process, if you'll see this process, here we have released EIF GDP. So this is inactive initiation factor 2. So this inactive initiation factor 2 needs to be activated. So for this activation, we require another factor that is called as 2B. So this 2B is chemically, it is guanosine exchange factor. It helps in the exchange of GTP to GTP. So these two combined together forms a complex which ultimately removes GDP here, this GDP, and adds GTP. So finally we are having the effective functional EIF2, and 2B will be separated and 2B will be reused for the another cycle. So ultimately we have got from the inactive to active factor 2, and this factor will be used for another cycle, another initiation process of the protein synthesis. So this is overall picture of the protein synthesis, where this is the first step where anti-association factor dissociates 40S and 60S. This 40S, this is our activation of tRNA. This activation of tRNA combines with this ternary complex, and ultimately this ternary complex combined with this 40S forms a 43S complex. This 43S binds with this messenger RNA to gives us 48S. Here tRNA moves along with RNA, look for the first AUG. Wherever it will find AUG, it will bind. Then comes 60S ribosomal components, and all the factors will be released, and ultimately we will have 80S. This 2F factor produces GTP, means has GTP, hydrolysis of GTP gives energy. Ultimately this will be inactivated and this will be recycled back for the active form. This is the initiation process. So initiation you have to understand in the sequence. For 4 steps we have seen first step was the dissociation. This one first step. Second step is 43S formation. Third step is 48S formation, and fourth step is 80S formation. Two intermediate things we have seen. One is ternary complex formation, another one is messenger RNA components. So if you remember all the things, it will sequentially you will easily revise it, you can remember it. So these are different steps, just to help you. Now these are the different factors, different, you can call it definitions. In the first step we had 40S. So these are two NPS with combined 40 and dissociate 60S. Then we had a binary complex where EIF2 combines with GTP, forms a binary complex. This binary complex combines with tRNA loaded with methionine. This is I means initiated methionine, and form a ternary complex. So ternary complex equal to IF2, GTP, and tRNA loaded on methionine loaded tRNA. 43S is this 40S 2 factor plus ternary complex. So 43S is ternary complex plus 40 plus 3 and 1A. Ternary complex means EIF2 GTP methionine. So that is this 43S. 48S is along with this 43S we are having messenger RNA and all the factors. So 43S messenger RNA and their factors. Similarly, we are having 80S where 48S plus 60S. So this is different definitions. Now we are going to see the elongation process of the protein synthesis, means third step of the protein synthesis. In this elongation step, we require certain factors, certain components. So the component which we required is the first is pre-initiation complex. This pre-initiation complex is nothing, it is the 80S complex from the previous cycle. Then we require tRNA loaded on the amino acid loaded tRNA for all other amino acids. We require 3 elongation factor similar to prokaryotic cells. In prokaryotic cells, we require other factors with other name. Here we require 1A, 1B, and 2. In case of prokaryotic cells, we require EF, TU, EF, TS, and EFG. Here TU is equal to 1A, TS is equal to 1B, and this is equal to 2. Same function, same thing will be done, only name has been changed. And this here energy source will be GTP. Similarly, this elongation occurs in 3 steps. First step is binding of amino acyl AMP, means second amino acid, to the A site, because first amino acid came, first methionine came and it binds to the P site. Now all other amino acid will come and binds to the A site. Second step is peptide bond formation, and third step will be the translocation. So we'll say elongation. In the elongation process, this is the first step. So this is from the previous initiation we have got 40S, 60S, and the first amino acid. Now new amino acid comes for the binding to the A site. This amino acid will not come alone, it will come along with some factors. Those factor is 1A, and but this 1A will not be alone, it will come with, it requires GTP. So this factor comes and it binds to the A site. Now peptide bond formation will occur. So this is called as transpeptidation reactions. This transpeptidation reaction is promoted by the geometry of peptide consisting solely the residue of the 28S ribosomal RNA. This 28S ribosomal part is the part of large ribosomal subunit, means this is part of 60S. So the transpeptidation reaction means peptide bond formation is assisted by a ribosomal RNA which behaves as enzyme, that is peptidyl transferase enzyme. This is 28S and this is part of large ribosomal subunit, that is 60S. Means this is not catalyzed by any enzyme, this is catalyzed by RNA. So this RNA is called as ribozyme. Ribozyme means any RNA which can utilized as, which works as enzyme is called as ribozyme. So this is called as ribozyme. Now in this transpeptidation reaction, this is the first amino acid we are having. This is second amino acid which came along with the factor 1 alpha and GTP. This transpeptidation reaction occurs. The second amino acid is having, this is our second amino acid and this is our first amino acid. So second amino acid is having nitrogen amino group. This amino group is having two lone pair of electrons. This lone pair of electrons attached on the carbon of the first amino acid present on the p site. So transpeptidation reaction is initiated as a nucleophilic attack by the lone pair of electron present on the nitrogen amino group of amino acid present on A site to the amino acid on the P site, and there will be a bond formation occurs. So this is called as peptide bond. Previous bond with the tRNA on the P site will be dissociated. A new bond, peptide bond, has been formed. So now we are having this type of situations where on the tRNA on the A site is having two amino acids and the tRNA on the P site does not have any amino acid. So this is a free amino acid. After that, this is again one of the example of how reaction occurs. So this is the amino group which nucleophilic attack on this carbon and a bond formation occurs and this bond will be dissociated. In this elongation reactions, then another factor is there, that is EF2, elongation factor 2, which do the translocation. So this is helping in the translocation reaction. Translocation means the movement of ribosome towards the 3' end. So our ribosomes moves one step further. Ultimately, what will happen? This tRNA which is at the P site will come to the E site. This tRNA which is at the A site will come to the A site, sorry, P site, and A site will be vacant. So as you are seeing here, this A site is vacant. The tRNA which was here, it came here, and the tRNA which was here, it came here, and this is E site. So it means this free tRNA exits. So that has gone. Here also, as we have seen in the previous steps, we have used a factor with the GTP, and this GTP will be hydrolyzed, gives so 1A becomes 1A GDP, means this is again inactive. For every new amino acid will come along with the tRNA, we require a fresh 1A with GTP. So this needs to be activated, and the activation will be similarly as we have activated that initiating factor, that is like this one. This 1A GDP will be activated with the help of 2B. There, sorry, 1B. There it was 2B. Here it is 1B, and the reaction is almost same. Ultimately, we will have 1A in activated form, and that will be reused for the different reactions. Yeah, for next cycle of the reaction. Then comes to the termination. In case of termination, there are three termination sequences we are having, that is UAG, UGA, and UAA. This termination sequence does not recognized by any tRNA, means we don't have any tRNA which is going to recognize this genetic code. So we don't have any anticodon for this one. So as soon as this any of the stop codon comes at the A site, so because we don't have any tRNA here, and rather we are having a releasing factor that is ERF1, which is structurally similar to the tRNA. So because we don't have any tRNA for this place, no anticodon recognized, and this RF can easily binds to this site. So ultimately we are having releasing factor binding to this site. This releasing factor uh, having a water molecule on the cell surface. Once this releasing factor binds there, this peptidyl transferase reaction occurs. So this peptidyl transferase reactions will be assisted by this RF3, releasing factor 3. So the peptide bond has been transferred to this RF1. RF1 was here, so RF1 got all the peptides by transpeptidation reactions. And then translocation will occur. So releasing factor along with all the things came at this place. And because water cannot hold peptides, so peptide will be released from there. The difference between the prokaryotic and eukaryotic is in case of prokaryotic, we had two releasing factor, RF1, 2, and 3. In our case, so two releasing factor in case of prokaryotic cells recognizes two different uh, anti means stop codons. In case of eukaryotic cells, we are having only one releasing factor, that is RF1, which recognizes all the three stop codons. So ultimately, this peptide will be released and protein synthesis has been done. All the factors has been released. So this is protein synthesis, all the three steps: initiation, elongation, termination, along with amino acid activation. Now we will see in this place what are the different factors we have used. We have used in case of elongation we have used 1A with GTP. We have used 1B for the recycling of this 1A, and factor 2. All these are E EF, means in eukaryotic elongation factor 1A, eukaryotic elongation factor 1B, eukaryotic elongation factor 2. This 2 will be translocation. This one is for every new amino acid will come on the tRNA with the help of this one. This is just to reactivate recycling of the 1A, and this is for translocation. Termination, we had having only one factor, that was eukaryotic releasing factor 1. We are having RF3 also, but this helps in the translocation reaction. In the initiation, we require a number of factors. Number of factors means we require factor number 1 to factor number 6. Factor number 1A and factor number 3 was anti-association component. This helps in the removal of dissociation of the ribosomal components for TS and 6TS. Then come to then come to factor 2. This factor 2 comes along with the first amino acid with the ternary complex. So this is factor 2 helps the ternary complex, and 2 will be for the recycling of this one. Factor 3 is again part of this anti-association complex. Factor 4, mainly factor A, factor G, 4A, 4T, 4E, associated with the cap portion. So they forms a cap binding proteins that is called 4F, binds to the messenger RNA cap. On 3' end, we are having protein poly A tail binding proteins. So this is 4. 4B helps in the association of formation of 48S. Then factor 5. Factor 5 was just helping in the formation of 80S complex. And factor C will be associated with 60S ribosomal components. So these are the different initiation factor, elongation factor, and termination factors. 1 to 6 we are using initiation factor, that is in initiation. Elongation we are having 3 factors, 1A, 1B, and 2. Termination we are having only 2 factors. Ultimately recognition will be done by only RF1. So that's all for the protein synthesis. In the uh, uh, presentation, next talk, we'll talk about the uh, post-translational modification and the differences between the prokaryotic and eukaryotic transcription process, and then we will talk about the inhibitors of prokaryotic and the eukaryotic translation process. That's all. If you have not subscribed my channel, please subscribe it. If you like the video, press the like button. Thank you very much. Have a nice day.